Thank you, James. So we are so excited for you all to hear more about what the third cohort of the Hoffman teams have been up to over the past year. You'll hear from an interdisciplinary group of faculty. So this really represents kind of that interdisciplinary nature that is HAI. We have folks here from all 7 of the Stanford schools, and they were selected from a highly competitive pool of applicants and also went through the Ethics and Society review, which looks at the ethical risks and benefits of the work to society more broadly. The teams received funding from this grant for 1 year initially, and over the next 2 days will be evaluated for additional 2 years worth of funding. Today, they are presenting to all of you, and then tomorrow they will be meeting with the selection committee for private interviews to dive a little bit deeper into the work and their goals over the next 2 years. Displayed, oh, next slide, I think. We have a slide highlighting our selection committee. So they're sitting right here at this front table, and they have a very hard job over the next 2 days choosing 3 of the 6 teams to win additional funding. So quick thank you to them for spending time with us and working on the Hockey team. And I'd also like to thank Christine Ravel, who is the research program manager, has been working really closely with me to making all of this happen over the past year and also the next 2 days. So before we introduce our first team, a few logistics. For Q&A, we're first going to give the selection committee an opportunity to ask questions after each team. If there's time after those questions, then we'll open it up to the audience here in the room, and we'll have mic runners. So raise your hand, and we'll bring a mic to you. So with that, I'm pleased to introduce our first team. They are titled A Multimodal Odyssey into the Human Mind, and this team is building a world model of the human brain. So the model promises to enhance diagnostic precision, inform personalized care, and deepen our understanding of neurological and cognitive processes. So let's welcome them to the stage.
Awesome. Perfect. Team Mind here. I'm actually very happy we are starting as this first presentation because now you're all part of an experiment. If you, at the end of this talk, are more awake and more inspired, we can just call it neural activation, which is the topic of our talk. So let's get started. I want to start with topic of foundation models, and I'm sure everybody here is familiar with it. Foundation models are large-scale AI models that are typically neural networks, but trained on extensive sets of data. And by virtue of their scale and data sets, diversity of data sets, they can solve many different tasks, including those that are not they are not trained for. Okay, so and foundation models was first coined by the team at CRFM, led by Percy, Rishi, and team, and many of our PIs are also part of that group. Today's most well-known AI models are foundation models. You know them all, from chatbots, conversational agents, content creation, and so on. They work 24/7, and they never get tired. They're always keeping their friendly tone. And beyond that, next up is image generation and video creation models that, again, we are all working with. There is also the artistic side of AI with music and music composition and so on. And now even generative AI is shaping the future of the future in ways that we are only beginning to understand. For example, drug discovery is creating human impact by creating new drugs. And even beyond that, we've been testing these models in very critical spaces, even ICU rooms, explaining what's happening at the bedside and even doing patient monitoring. And not just that, creating extensive and detailed reports. So, uh, with that, I want to talk about the brain and what a brain foundation model is. We don't have such a model. All of these stories that I just told about the brain and brain foundation models, but a brain foundation model could be a digital twin of the human um, brain, which includes not only the knowledge of brain structure and function, it also encompasses the phenotypic and genotypic aspects of human health. And such a system can be used for, say, clinical diagnosis in unprecedented ways and could be used for designing new interventions, new treatments, and so on, which we'll be talking about today. But it's specifically important to create such a model because it can help accelerating fundamental research in neuroscience discovery, just because the model can simulate neural responses at scale, and it can also um, help with transforming AI, robotics, and so on, because again, we are modeling human brain. But I want to highlight that this type of technology is most importantly useful because they can enable precision diagnostics and personalized treatments. Such models could generate disease-specific neural or behavioral signatures, or could be used for early detection of neurological psychiatric disorders, and could be used to optimize neural interventions through in silico experimentation. But in order to do so, we need to first perceive the brain, and how it is done is through medical imaging, different modalities from brain structure, which we often use MRIs and diffusion tensor imaging frameworks, and to measure a function, we often use functional MRIs, fMRIs, and other types of signal data. So with that and these other aspects of genotypic and phenotypic data, which we are actually in reference to physiological and pathological aging and neurodevelopmental mechanisms, we've identified these different aspects that contribute to human brain health, from psychological, biological, environmental, and physical factors interrelated to the health, and the type of applications. To build a foundation model, and now actually let's call it a brain world model, or BWM, because it encompasses beyond just modalities of imaging in the brain. We need to create a generative process similar to many other applications. So we use all of the data that I described as the input to the model, and then same data, say, as the output. And here we want to learn a shared representation. In most of the biomedical applications, we often focus on resting state functional MRIs, but in neuroscience, in many neuroscience applications, we often look at task-based functional MRIs, where the goal is to decode or understand the neural processes of the brain with respect to any input stimuli. So with that also included, we now can say, with this model and all of this vast array of data types, we will train the model, as you can see with that tiny animation there. But the very first challenge is on what data. So data is the major challenge with these types of projects. And to this end, we collected, curated, and pre-processed a large set of data from public and private resources with over 150,000 unique participants with longitudinal imaging data and at least one of the other modalities. And the age range spans from 5 to 100, 5 to 95. So we created a unified pipeline, publicly released code and pre-processed data for all of the vast majority of different types of imaging and non-imaging data with extensive QC reports and so on. And in order to further be able to harmonize the data within the models, because again, data is coming from vast different sites, different parts of the world even, we have developed a learning-based harmonization technique in which it's basically a simple neural network layer, metadata normalization, which could actually be used very much similar to batch normalization in any architecture, and harmonize the data, remove the shifts in distribution in the features in each architecture. And then this is done by just running a very simple generalized linear model. And what we've done in a recent paper in ICML 2025 was extending this to novel architectures, continual learning, and so on. So after partially solving the problem with data, the next step is to do training and model training. What we observed and what we've been proposing was trying to train the data with any combination of available data in the data sets in terms of the different modalities. And therefore, we can create multiple training pathways. One of those is, for example, single modality training pathway, which is the current practice in most foundation models, brain foundation models even. They use reconstruction, masked autoencoding, and so on as training objectives. We could do the same with other modalities, but then based on the availability of data, as I said, we can train the models with multiple modalities or just train across different modalities, cross-modality pathways, such as decoding the stimuli from brain function, or even decoding physical function improvements, impairments from brain data, brain function, brain imaging data. Another pathway is that is common in many data sets includes linking genetics and genotypic and phenotypic data to neural structures and functions. So, as I said, we could simply train the models with any combination of data available. And let's look at some few examples that we've completed in the past year. We recently published a generative model called MorphLDM, Morphological Latent Diffusion Model, which unlike previous methods that often try to generate the brain MRI at once, we argue that human brains often follow a template. Your brains are very similar to each other and have a lot of structural similarities. So with that, instead of generating the image from scratch, we started by generating deformation fields, which could actually be added to the template to generate the actual image. So we built this on top of LDM, a latent diffusion model, in which an autoencoder is trained and a diffusion process operates in the latent space of the LDMs. And to generate the new image, you could just sample a point and then run the process. MorphLDM does a similar process, but instead generates a deformation field, which is then added to the template that is also learned from the data itself. And to generate novel images, again, we just need to sample a random point and decode the deformation field and so on. So since MorphLDM doesn't begin from pure noise, but instead builds upon a learned brain template, the denoising process refines realistic brain details rather than generating from scratch as traditional methods like LDM does. And this approach makes the generation process significantly more robust, and it incorporates strong structural priors and about how the brain should look. And some of the examples and how the quality of the newly generated images look like. And not just that, fidelity and loyalty to different factors, biological factors, for example, age. You can see between real and our MorphLDM methods. Our method was able to do much better generation in different age buckets and be closer to real distributions. Along the same line, our colleague Akshay and team have trained MedVAE, a variational autoencoder on medical imaging data, which is not only using 2D images, but it was trained on a large group, nearly 32,000 3D brain images, volumetric images as well. In addition, we actually we looked into evaluation metrics, and in the brain sciences and in general anatomical structures, we don't really have very good metrics to evaluate generation. Everybody's still using FIDs, which is mostly used in computer vision, but we've shown that they are not good metrics, and all the other MMD and SSIM metrics, and instead we proposed Wasabi, which is a metric for evaluating anatomical plausibility of brain images. And as you can see, it can do a very good ranking of comparisons, healthy versus healthy, having the least amount of difference, and then between a healthy and somebody with cognitive impairment, sex differences, and even those with Alzheimer's disease, we can see a very good ranking, which shows this metric is a great metric. So we've looked at some of the models, and there are way more applications, a lot more different models that we've developed, but I'm not going to go through all of them. Briefly, I want to talk about some of the applications we've been working on and how we define now, instead of training pathways, some of the inference pathways that we've worked through. So finding the inference pathways can help us look into different applications, including, for example, this one, which is around neural decoding and decoding the stimuli from the brain function. What I mean is, we worked on this paper with our colleagues where we wanted to decode what the person is seeing while they are in the functional MRI machine. And we used our models, our frameworks to do that. And in order to do this task, we trained fMRI, image, and text encoders together to have a learned representation space, and then the training decodes the images through diffusion processes. And during testing, we only use the fMRI signals and decode the image itself. Compared to some of the previous works in this space, MindEye and MindViz, qualitatively our results are significant, but not just that, even quantitatively, we could prove that the generated images that our model had is more loyal to the data, the stimuli. So in the next work, it's under review. We also did the same by modeling the process as a neurosymbolic reasoning framework. And we showed on these two data sets that we see the predicates, the concepts naturally ground to brain regions that their constituent objects are grounded to. So a compositional framework for mapping concepts to brain regions and their activations. So as a next inference pathway and application, I want to talk about this inference pathway between brain function and an action, which as an application, it can have robot learning applications, controlling a robot arm with the brain. And how we've done that was through EEG signals using same type of foundation modeling that outputs the actions, and those actions can be used to control the robot arm. And this is an example done by our colleagues at SVL, where EEGs are being used for controlling robot arms. And beyond applications in neuroscience discovery, there are quite a few in biomedical discovery, and we call this diagnosis inference pathways that many different types of data could be used for diagnosis. What it helps is not just the diagnosis itself, a binary diagnosis. What we can get from it is also generating or extracting digital discovery of biomarkers. Another way of doing the same type of digital biomarker discovery is through relating function and neural relationships. And that's what we've done in a few projects. And we've done this data-driven discovery of movement-linked heterogeneity in neurodegenerative disorders, paper published recently in Nature Machine Intelligence, where we use same type of motion encoders, action movements, and functional MRIs to create a representation space which is common between them. And that means we've been identifying some digital markers only from video of these movements that highly correlate with brain impairments, brain disruptions, which means they're actually very good proxy of brain disorders. And this means we could actually turn them into, say, a mobile app that can easily be deployed over time. We can generate a lot of longitudinal data and measurements of the brain function through this type of analysis. It's scalable, repeatable over time, and so on. So with that, in terms of biomedical discovery applications, there are quite a few other ones, for example, genotype, phenotype, inference pathways, which I invite my colleague Hanschel to continue.
So yeah, as you know, genetics influences a whole variety of human traits and diseases, including brain development, activity, aging, as well as disease. So we are very excited to work on what we are referring to as the variant to function to phenotype platform, V2F2P. The goal here is to decode the genetic, molecular, and cellular basis of brain phenotypes and disorders by leveraging these brain foundation models. And to do so, we are integrating data across multiple scales of human biology. So ranging from population scale sequencing of individuals available in large biobanks. So this allows us to catalog genetic variation across tens of thousands and millions of individuals. Many of these biobanks pair this genetic data with extensive patient phenotyping that includes clinical data, brain imaging modalities, and so forth. But the missing link between the genotype and phenotype is how these genetic variants influence phenotype through molecular activity, right? Which cell types, which genes, which genetic variants, which molecular pathways, how do they influence brain phenotype? That's the question we're trying to answer. And I'll briefly walk you through the strategy of how we're doing this. So leveraging these brain foundation models, we can take brain imaging datasets from these biobanks, and we can use our foundation models to embed these brain imaging datasets, thereby extracting rich features and phenotypes from the data. Now, given this information, remember that in these biobanks, these images are paired with genetic information. So we can use classical statistical genetics methods to link and map genetic loci associated with all of these diverse brain phenotypes. This has been widely done for disease phenotypes. So, for example, you can take patients that have Alzheimer's disease versus control individuals and perform these kinds of statistical associations. And they reveal, as you can see the figure on the right, that's called a Manhattan plot. The x-axis is the genome, the y-axis is the significance of association of loci. Each of those little points is a genetic variant, and these towers, the Manhattan towers, are basically loci that are strongly associated with the disease. But you can apply this same strategy to not just disease and no disease classifications, but to quantitative brain phenotypes. That's what we're planning to do. Now, once you map these loci, they kind of give you approximate genomic regions associated with the disease, but they don't tell you the causal mutations, the genes, the molecular networks, and the cell types that are mediating the effects of genetics on these phenotypes. So to do so, we're going to leverage another class of deep learning and machine learning models. These are referred to as sequence-to-function models. These are molecular models, and what they do, think of them as text-to-speech converters. The text in this case is the human genome, the DNA sequence, and the speech in this case is hundreds and thousands of molecular profiles measured across the genome in thousands and thousands of diverse cell types and tissues, often at single cell resolution. So we can train these neural networks that act as these translators, mapping DNA sequence to molecular activity. And then we can take candidate mutations in the genome, push them to the model, and the model can make predictions, as you can see at the bottom right. You take a genetic variant, healthy individuals have an A at a position, disease individuals might have a G, and you can in silico predict the effect of this mutation on molecular activity. And using XAI techniques, you can also interpret how the model's making these predictions, thereby mapping causal variants, causal genes, and likely cell types through which these variants are acting. And we've already trained these models at scale over thousands of diverse cell types and cell states, from fetal brains, adult brains, and brain data from diverse neurological diseases. And finally, we can take these predictions, we can integrate them with genetic and molecular networks associated directly with disease, thereby developing novel integrative scores, risk scores for patients. And finally, we can take these variants and validate them in the lab. As you can see on the right, this little image of a mouse embryo. We can put these variants with the two different variations into these mouse embryos or mouse models or iPSC-derived brain cell types to validate their effects. So this is something we're actively working on over the next year. I'll now pass it on to Dan.
All right, thank you. Okay, so so far we've heard a lot about what and why we're doing making brain foundation models. But one of the key things that we want to do with them is build interventions, right? And what are interventions? Interventions are sort of the idea, the core goal being to use the model to figure out how to change brain states optimally to achieve desired behavioral change. Okay, that's a sort of core, like kind of key long-term thing we actually want to really do with these kinds of models, whether it's through genetic pathways of inference or whether it's functional pathways of inference or all kinds of different pathways of inference, right? So I'm going to tell you a little bit about how that actually works so it's sort of concrete in your mind. Okay, so the one thing, so first thing is to realize is what are the kinds of intervention devices that we can have. So two very promising types of intervention devices that we have right now are transcranial magnetic stimulation and focused ultrasound. These are basically brain devices that you can use to focus little changes in the brain as you need to. Okay, and of course, there's also direct behavioral intervention. So basically giving people some kind of behavioral therapy effectively to get them to change their choices in particular situations. So these are key kinds of intervention machineries. Okay, they're kind of hardware. Okay, so the way to think about the way we think of the model in this context is as the software for driving those intervention devices. Okay, so for example, let's say you have an output effectively of a certain kind of behavior that you'd like to change, like for example, being in a depressed state. Okay, that's in the behavioral context at the end of the day. However, of course, because behavior is largely influenced by what's going on in the brain, there's a brain state behind that depressed state, right? So there's like effectively a depressed brain state that's underlying that. Okay. Okay, but luckily, by talking in virtue of the kind of diagnostic tools that Asan mentioned earlier, we also have a characterization of what counts as a healthy state. Okay, so it's a healthy state and a depressed state, right? So now, how do we create that and use that information with the model to create an intervention? Okay, effectively, you have some kind of intervention device. Let's say it's TMS. That's what's used for depression in a lot of cases, right? So what you want to do is you want to use that intervention device to like do something to the brain that's targeted to change the state, right? Okay, so the core thing is you can take the model and invert it effectively to figure out how to drive the system to reduce the distance between the depressed and the healthy state. So this is effectively one of the key techniques in machine learning neural networks is backpropagation. Because we have the internal structure of the system, we can use that to reverse the inference of the model back to what you need to do on the intervention device. So basically, you want to minimize the difference between these two things. We want to optimize the target objective of driving this to zero effectively, right? And suppose we are able to do that, and we're able to do that in many existing kind of contexts right now in brain data and otherwise, then what ends up happening is that we can take away the intervention device. The healthy brain state is what has arisen. And of course, if you push that forward through the inference path of the model or of the real brain, that should change the behavioral state to being what you would like that to be. Okay, this is the core mechanism of building an intervention. Okay, and so that's underlying a lot of our thinking and a lot of the ways that we plan to use these kinds of models. Okay, so let me tell you very briefly about two examples of doing this. So this is back to the neural signal operated robots, and in particular, the idea that you can use this model to drive brain-machine interfaces. So here's an example of essentially like test time optimization to figure out how to build interventions that will achieve goals as desired. In this case, it's a person playing a keyboard with their mind effectively, and that can also be used to do things like pick up complex objects and do actual things that you wish to do by pushing that data through the model. Okay, this is an example of the same kind of system that Ehsan mentioned earlier, but now turned into a clinical objective. Okay, there's another form of intervention that is very much on our mind, which is direct kind of cognitive causal discovery. And this is via the notion of using this sort of model as a brain digital twin. And so the key notion, though, is that we can make interventions in the lab right now. Okay, but the question is, how effective are they and where are they changing the brain, right? So in particular, you can do before an intervention, you have one brain state. After the intervention, you have a different brain state. What has changed and what has the impact of that been? And you really would like to be able to make predictions about which interventions are effective. So this is work from Van Key and Lin and others on the team. And basically what they have done is they've taken examples of interventions on trying to improve episodic memory performance in adults with mild cognitive impairment. And the key question is, where in the brain are the changes that the intervention's happening, right? And it turns out that if you build a brain model of the kind that I've told you about, you can use that to make decent predictions of where that is happening. So those are the high bars toward the right in those bar plots. Those are effectively the fact that you can use the models to understand the effect of the intervention and then go towards building more effective interventions by backpropping through the model to achieve the sort of core goals. Okay, so I'm going to turn it over to Ehsan to finish up now. Thanks.
Awesome, thank you. Yes, to close the presentation, to finish the presentation. So far, we've been doing a lot of work on model training, as I presented some of those, and applications in neuroscience and biomedical discovery, but there is a lot of work to be continued. Before saying what's next, I think I wanted to say that next step for us is also to add language to this mix, to just turn this entire system as a neuroscience agent model, so we can explain all of those components that we've talked about in plain language. But beyond that, what our next steps are is for sure, scaling up data and training and multimodal brain training and so on. But what this gives us is in the next 2 years, we will have two major products. Genotype, phenotype informed digital twin of the brain and a platform for building automated intervention and treatment design. And going back to all the way back to what we started with and why we need these models, this enables precision diagnostics, personalized, optimized treatment, and also gives us an agent model that can explain everything. To do this, we've built a huge community. We had weekly meetings, 32 speakers, the list is here. What we've done was publishing a huge data set, releasing software packages, and writing 27 papers, some accepted, some submitted, and some in preparation. Our amazing team of PIs and research scientists, that's what we started with, research scientists, postdocs, and students. But now we also have, over the year, new members joining our group with their labs, with their expertise in human machine interface, brain computer interface, and so on, and intervention. Thank you very much.
We have a bit of time for questions, so if our speakers could come up here, feel free to move the chairs around if they're like too close. But yeah, feel free to sit wherever you'd like. We will start with questions from our selection committee. You all have a mic at your table. We'll start with Eric.
Yeah, thanks. What a nice constellation of related projects that came out of some of since the last 12 months. Seems like a great progress. Can you say a bit about the engagement with neurologists who could help guide some of the technologies at potentially valuable clinical results and interventions and mitigations and studies? I said I didn't see neurology represented anywhere in your team.
Probably, I can answer this question. So actually, all this intervention work we have done so far, so neurologists and neuropsychologists are always part of the team. It's just in terms of this particular team here, we may not have them, but in terms of the work we have done so far, like those cognitive training, because in order to do the deep phenotyping of the Alzheimer's disease and mild cognitive impairment, we definitely need this neurologist input. However, I'm not sure everyone here have heard the term in terms of diagnosing this disease. Traditionally, we have been using this, we call the consensus diagnostic process, which kind of subjective to some extent. So we actually are using this larger foundation model trying to help more objectively make deep phenotyping and also to better predict the kind of the improvement or effect from this intervention, which we consider as complementary to the diagnostic and the intervention measurement based on battery test.
And I want to just very quickly add that this project made us and helped us to start even new collaborations with folks in the Wuxi Neuroscience Institute. And we are representing that institute as well, but those who are running those intervention labs. And we've been experimenting, designing new projects based on those collaborations that are created, again, because of this project.
So I have a question about where you're at versus the goal. So clearly some of your earlier papers are, a model with a certain kind of data. When I think of the foundation model, I think of your initial slide of all this different data that can do all these tasks. Is the goal, is that achievable, that you would create one big model that has all of this and that's what you're doing? Or do you think it will still be different specialized models for the things that you want to do?
Yeah, that's a great question. I can probably start, and then Dan, if you have any thoughts, I would love to hear that too. Short answer to that is yes. The goal is in the next 2 years, we'll be creating that big model that I've talked about. And that's how we can create that digital twin of the brain and have the intervention, because intervention, digital twin, biomedical discovery is all an application of that model after it's built. So that's the short answer. But I also want to very briefly mention that in the past year, we had this huge challenge of data, which is now more or less out of the way. And now it's time to do this hugely and massively multimodal training. So hope that answers the question. And is there anything anybody wants to add?
Just a quick follow-up. How will you go about getting that massive amount of behavioral data?
Many of these
Not just the fMRI stuff. Like,
yeah.
I assume behavior. I want to know what's going on in somebody's everyday life.
So some of the studies that we've listed, UK Biobank, all of us, and some other internal data that we've created, our collaborators at ADRC, for example, Alzheimer's Disease Research Center, and other institutes, we've been collecting these data internally, and we have access to other types of data from these public data sets. Again, that's why we've been building all of these harmonization techniques, even learning-based harmonization techniques, and so on.
Yeah, and also in terms of intervention data, local data, which we have over 10 trials, clinical trials actually funded by NIH, and also nationwide, this public data set, including intervention, they always have behavior and the brain imaging related. Yeah, so it will, the behaviors are often ecologically natural behavior. So those are really valuable data for validation at least.
Thank you so much for a wonderful presentation and this exciting work. As a clinician, I have a question analogous to Eric's, which is if you're focused on interventions, you're focused on disease states and behavioral change, how are you planning to, in the next year, integrate your research with real people, patients? And I guess that relates to involvement of clinicians, which I wondered how you're thinking of it.
Yeah, so this is right on our target. So in terms of answering this, so two things we are trying to do. One is deep phenotyping using the multimodality foundation model. We will be able to complement what we consider as consensus diagnosis of because like currently the different kind of dementia due to AD, due to mixed dementia, due to different kind of pathology, and plus all this behavior, so executive function oriented, memory oriented. So all these are very complicated and can be very heterogeneous among older person. So a foundation model can definitely help with this kind of more systematic phenotyping. But on the other hand, in terms of intervention, we have been thinking two ways. One is we can learn, so using foundation model, it's very important to identify the hidden, we call the hidden brain patterns that are associated with some of the trials actually failed currently. And it can help us inform new therapeutic target. And then on the other hand, we are working, so Isa and I have been working a lot on closed loop human-machine interface. So a lot of this foundation model related work can be incorporated in there. And it's actually two of our current NIH funded project trying to do is incorporating this. So hopefully, phenotyping, diagnosis, phenotyping, intervention mechanism, and intervention process. Three things we can do with intervention. Yeah.
Well, let me add one thing to that, which is that there's also a tool building aspect of it. So if you could achieve interventions that like push behavior around in some kind of non-clinical but noticeable way with the tool, then when you come to the time when you have the clinical intervention, then you're really in good shape. And so what's been happening over the past few years, what's happening now is a lot of that work. So basically showing that if you have a model of the healthy adult, then you can like achieve some behavioral change goal within the space that is available in that population. The same tool can then be applied over in the clinical side, and we're in some part of the work is tool sharpening to get us there. So we probably have another year at least of that as well. But between the clinical efforts and the like tool building in the healthy adult sort of neurotypical situation cases, we hope to be in a position where we can put those two together and have a meaningful outcome.
Exactly. And just to add to that, again, the project was able to help us work with, say, for example, Dr. Corey Keller, who's running brain stimulation labs in Busan Neuroscience Institute and is actually now part of this project. So this is a community building. And as Dan said, I think in the near future, as the tools are built, we'll get more into that.
I would like to go back to James's earlier question about dream of building foundation models. Really super impressive, diverse range of work that you presented seem to suggest that if we have a foundation model encompassing all of this, it could be super powerful. But on the other hand, the modalities and the types of signals seem fairly distant from each other compared to the kind of foundation model that's built for language domain, and how even for video generation model, usually it's completely disconnected from language-based foundation model. And then the moment you try to build a foundation model, even interleaving language and images, there are different architectures to be developed. Something like transfusion or chameleon and a mixture of transformers. I don't know whether you thought about, what sort of a foundation architecture you might use or might need to newly design, and whether, relative to how LLM foundation model has been developed, to follow the recipe of a pre-training and then post-training and test time scaling, I wonder whether you see plans going forward on that note as well, because just a pre-trained model may not do very much unless you align it.
Yes, absolutely. That's a very smart and also timely question. Because of time, we didn't go into the technical details of how each of these are being achieved. But in a nutshell, we've been looking into the emerging areas of, as you said, mixture of experts, and also the tokenization of different modalities to bring them into a space that a huge model can make sense of them all together. It's very much similar to having language fed into a transformer as language, and all other modalities being fed to the same model as foreign languages, not English anymore, foreign languages. But that's how this is happening, is through tokenization, tokenizing everything, and through positional encoding and so on, we can identify their different types. But that's a very good question. As I said, due to time, we didn't go into very much of details, but that's the approach we are taking.
And you can just think, there's basically two types of data we deal with: image-like data and sequence-like data. The stimuli to the models and the brain data are actually image-like. So those go together really well. And then like the genotype, phenotype, behavior data, that's more like sequence data, right? So in many ways, those two things do fit into the sort of like modern synthesis. So I don't think architecturally we have to like invent like the totally new wheel to put those two things together. So there are some nice conveniences in the data structures that allow us to do pretty reasonable things. So we can talk, in the closed session in much more technical detail if that's really helpful.
So I had a follow up to the part about sharpening tools. Is the idea, like Dan, when you were saying, hey, somebody's depressed brain looks like this, and we want to figure out how to get to that state, is the idea that that would help guide changing like how TMS or things like that are done to help get to that
Yeah,
state?
exactly. So basically TMS is this honking thing that
Right.
people do right now. They hit DLPFC like with this huge hammer, and actually they can make the device a lot more focal than it is in the clinic right now. They just don't know what to do with the focality, right? So our thing is our core goal, or at least one of them, is to make the software to drive that focality. And it's easy to imagine, if you had the model, how you'd get the command to the TMS device. You basically push back through the model to the input on the TMS side, what pulse sequence at what amount of time and for how long and where exactly. And if you had the actual model of the brain, then that becomes like a fairly straightforward machine learning problem, right? So it's software effectively for driving hardware that is like pretty good right now. People just don't know how to use it, essentially.
And it's very effective, just needs to be optimized.
So maybe going back to discussions we've had in the past, Daniel, but also maybe asking Asan, but anybody else can join in as well. What would you say personally for each of you is a key mystery you think is being unlocked to date, or your aspiration, a key mystery about human cognition beyond the engineering and the applications you're pursuing through these tools and methods? Just one even comes to mind, top of mind right now for each of you.
I don't know if you want me to answer this question because I'm like an anti-mysterion. Okay. So I actually kind of don't think of the brain as mysterious. I think of it as an engineering box and I want to fix it and use it. And so it's hard for me to dream that way. I want to dream in terms of, like the guidance device for the TMS control system,
Okay,
essentially.
that's good enough for an answer. How about you, Asan? Maybe you're a dreamer, more of a dreamer.
That's
Maybe
it.
you believe that there are mysteries.
Probably. So I'm also an engineer. So I'm looking at this from an engineering perspective, trying to create the building blocks that makes sense of what's happening. And as you saw throughout the entire presentation, we are putting these small building blocks to make sense of brain function, structure, anatomical aspects of the brain, and so on. Even our methods are designed from that perspective. And the ultimate goal is to translate that technology, that tool into real application. It's not just tool building here. We are looking into the application itself. So again, that's an engineering perspective, I admit.
The scientists will use it. If the brain model exists, some scientists are going to take it and run overnight, like millions of experiments that otherwise would have taken them, hundreds of years, and that will help them unlock the mysteries that they're so excited about. So,
and just to
Okay.
be clear, Daniel, I mean, go back. I don't want to debate in front of the whole audience right now, but
we've had this discussion before.
there likely are principles to be unlocked that are not all just tiny little hacks. Anyway,
Yes,
comment.
great.
Can I just chime in one thing very quickly? So just in terms of like cognition, so as a clinician here, so I've been thinking in terms of abnormal versus normal cognition and abnormal versus normal brain for a long time. And just through this foundation model project, actually, I start thinking, so what is considered generative and what considered specific? Actually, there is no like very clear boundary here. So go back to the question about whether eventually we should build a multimodality brain imaging model that will cover everything, or we should do more specific development of selecting certain modality. Actually, I think it's a very good, very important scientific question. We should try to do benchmark because, so far, we see some of the signals brain function-wise may be more generative, but in terms of brain structure, actually, they may be actually more specific in terms of pathology-oriented. So I think those are actually in the work we will be doing for the next step. Yeah.
Related to the benchmark comment, I think we need quantitative markers essentially to really understand what's going on in the brain. And I'm coming from the psychiatry perspective. And so what I see a lot, it's very subjective. And I hope with these foundational models that we are developing, that we can come to these quantitative markers so we can build benchmarks so that we then are able to better understand kind of what's going on in the brain, and specifically with respect to diseases.
All right. With that, we're going to close out this session. Let's give our team a round of applause. Thank you so much.